DNA replication is the process of a DNA molecule making a copy of itself. This takes place during cell division. Remember, the job of DNA is to pass genetic information from parent to offspring. So during cell division, the DNA molecule needs to copy itself so that the information can be passed on to the new cell. So here's kind of a schematic of the DNA replication process. The original strand of DNA is copied, and if you see down here in the product, these are going to be the two strands that result from the replication process. Each one of these new strands, if we want to call them that, contains one of the original strand from the DNA, which I'm showing in pink. And then also each one contains a new strand, which I'm showing in blue. So the strands that result, the resulting DNA, I don't really want to call it new because it's half new and half old. The resulting DNA is one half or one strand of original DNA, and it is one strand of new. When the DNA molecule replicates itself, it actually just kind of opens itself up a little bit at a time from the bottom up and just makes actual copies. So I'm going to attempt to kind of draw what that might look like over here. I'm not a very good artist, but I'm going to draw like an unzipped DNA strand. That's the word that we use, and say this is the 3' end of one strand and this is the 5' end of another strand. So this would be the 5' end and this would be the 3' end. And so the DNA molecule just starts to break these hydrogen bonds down at the bottom to open up and separate the individual strands of DNA. The new strands of DNA are synthesized alongside these original strands, just to actually be really consistent. I'm going to change this to be colored pink so it matches the same coloring I'm using over here. So the new strands of DNA are going to be synthesized here alongside this DNA molecule as it opens itself up. So we'll get one strand synthesizing right alongside here, and as this new strand is synthesized, the hydrogen bonds will connect it to the old strand, and then it will also begin to twist itself up into the double helix shape. This new strand is being synthesized from its 5' end to its 3' end, and this new strand is being synthesized continuously. And I'm going to elaborate on that in just a second. Continuously. I'm not sure if I spelled that right. So this is a continuous synthesis of this DNA strand. The DNA molecule can only synthesize itself in this exact order here, from the new 5' to the new 3', or sometimes it's described as synthesizing from the 3' to the 5'. It really depends on which strand is your frame of reference. So when we're looking at this synthesis, if it can only synthesize from the 5' to the 3', well, what's going to happen over here with this strand? If we can only synthesize from the 5' to the 3', that means that this strand has to synthesize itself in this direction, which is kind of backwards, in terms of how this DNA molecule is uncoiling itself. And what we see is that as the DNA molecule uncoils itself, this side is just synthesized continuously as the strand opens up. But the other side can't synthesize itself continuously. It has to open up a little bit, and then it's able to do a short segment of synthesis from 3' to 5'. And then it has to wait for the DNA strand to open up a little bit more, and it can synthesize a second piece also from 3' to 5'. And this is a discontinuous synthesis of DNA taking place over here. As these individual chunks are being synthesized, there is another enzyme that will come in and connect these together, so it won't stay broken. It will all eventually be one continuous strand. It just can't be synthesized in continuous order. The strand that is synthesized continuously is often referred to as the leading strand because it typically is synthesized faster than the other strand, which is referred to as the lagging strand. Because again, remember, it has to uncoil the DNA a little bit before it's able to actually synthesize that strand of nucleotides.